Hello everyone, I will discuss the drug discovery stages and modern advancement. Drug discovery is a multi-step process that begins with target identification, where scientists determine the biological causes of disease and select a suitable target. The ideal target should be safe, druggable, efficacious, and meet clinical and commercial requirements. Each stage narrowed down candidates from thousands of molecules to one validated target as the foundation for all later drug development steps. Next is target validation, which confirms that this target truly plays a role in disease using various biological and molecular techniques such as SAR analysis and downstream signaling monitoring. Each stage building greater confidence that the target is both the right one and the safe one to pursue before investing in the full drug development. Then comes lead identification, where potential drug molecules are discovered and evaluated based on their activity, stability, and ability to interact with the target. It will relate the structure-activity relationship, assessing whether it can be synthesis and gathering initial evidence that it works in living system and engage its target. This step filtering thousands of compounds down to a select few with the best balance of activity, safety, and real-world viability inside the body. After that, lead optimization improve this molecule through structural modification to enhance their pharmacokinetic, pharmacodynamic, and safety profile. It conducted through repeated modification cycle and testing where each round will improve the structure. Lead optimization turns a promising molecule into a refined and tested and ready preclinical drug candidate. Once it's optimized, the drug undergoes product characterization and formulation, where its physical and chemical properties are studied and the base dosage form is developed. The parameters evaluated during formulation, such as solubility in different media and solvent, it ensures that a chemically effective drug is also physically deliverable, where it transforms the optimized compound into safe, stable, and patient-ready medicine. This is followed by preclinical research where the drug is tested in animals to assess the safety and efficacy before human trial. It can be conducted in two ways, general pharmacology and toxicology. Pharmacology deals with the pharmacokinetic and pharmacodynamic parameters, while toxicological studies can be performed by in vitro and in vivo tests. This will ensure the drug works as intended and is not dangerously harmful before it is administered to a human being. Next, an investigational new drug application or IND is then submitted to regulatory authorities to begin the clinical testing. This step ensures that every approved drug remains safe, effective, and properly guided throughout its entire lifespan on the market. The drug then enter the clinical trials conducted in phase 1 to 4 to evaluate the safety, dosage, and effectiveness in humans. This is the most critical stage where the drug's true performance in humans is established, forming the foundation for regulatory approval and eventual patient use. Finally, a new drug application is reviewed by FDA and submitted for approval. If the drug is approved, the drug is being labeled and then marketed and continuously monitored for safety. This step will ensure that every approved drug remains safe, effective, and properly guided throughout its entire lifespan on the market. First, academic-industrial partnership is very important. This collaboration helps tackle difficult and druggable targets and reduce drug failure rates through data sharing. For example, Academic Drug Discovery Consortium (ADDC) initiatives formed to share resources like compound libraries and biological assays and bioinformatic resources. Other than that, computational tools and bioinformatics are also widely used in pharmacological research. Scientists can now predict ADMET properties, perform molecular docking, for example via X-ray crystal structures, and then biological target and their ligand provided by FDA Orange Book and Drug Bank help to identify potential drug candidates more efficiently. Lastly, this slide shows how modern drug design uses key rule and metric. Lipid's key rule of 5 ensure good drug absorption, while the additional parameter will refine the drug properties. Tools like matching molecular pair analysis help predict how small changes affect the performance. Thus, this will improve the overall drug development success. In conclusion, modern advancements have made drug discovery faster, more efficient, and more collaborative, increasing the chance of developing safe and effective drugs.